Hello and welcome all. My name is Karl Nyman and I'm an international student originally from Sweden, but currently studying my bachelor's degree in biomedical and health science at St. Leo University campus in Florida. In 2020, I received a science scholarship from the National Honor Society of Tribera, and I used the funds to partially fund my research project that I will present for you today. My project converged around comparing the cytotoxic effect of a well-established chemotherapy drug used for pancreatic cancer treatment in clinics today, gemcitabine, with a derivative of the prominent tumor suppressor retinoblastoma. Pancreatic cancer is extremely deadly and kills almost 50,000 people in the United States each year. The 5-year survival rate for someone diagnosed with pancreatic cancer is as low as 9%. I've personally been affected by this horrible disease as my grandfather passed away with pancreatic cancer before I was born. The current viable treatment methods for pancreatic cancer include surgery, radiation, chemotherapy, chemoradiation, and targeted therapies. All these treatment methods have severe side effects in one way or another. Surgery can lead to blood loss and does not eradicate metastasized cancers once it has spread in the body. Chemotherapy, however, can be used for metastasized cancer, but negatively affects all cells in the body, leading to severe side effects such as anemia, immune suppression, and hair loss. There's certainly a need for a novel cancer therapy that can eliminate the cancer cells without interfering with normal cells. Gemcitabine is a well-established chemotherapy medication used as a single agent for treatment of pancreatic cancer. The structure of gemcitabine, just like many other chemotherapies, resembles an intermediate in metabolism and is picked up by certain nucleoside transporters of the cell that think that the chemical is a normal intermediate of the cell, and therefore it's transferred over the cell membrane. Inside the cell, gemcitabine is converted to its active drug metabolites that gets incorporated into the DNA by, once again, an enzyme that thinks that these metabolites are just like a normal nucleotide. And after the addition of one more nucleotide to this gemcitabine metabolite, the termination of DNA machinery occurs because of a sterical problem due to the size and structural differences of this gemcitabine metabolite that has been incorporated into the DNA. And this leads to a DNA single-strand breakage as DNA repair mechanisms are unable to locate the problem, and the apoptosis and cell death occurs. Gemcitabine also has other modes of inhibition, including a self-potentiating mechanism, making the cells more susceptible to taking up more of the chemical than its normal counterpart. Worth noting is that gemcitabine is not cancer cell specific and therefore equally inhibits DNA replication for all cells in the body. RB peptide 7MM is a derivative of the tumor-suppressing protein retinoblastoma. Retinoblastoma protein is a naturally occurring protein in the body, preventing excessive cell growth by directly inhibiting the CMG helicase, a key component of the cellular replication machinery in DNA replication. There's evidence suggesting that cancer cells acquire a reduced number of dormant or resting CMG helicases compared to normal cells. Hence, when targeting the CMG helicase for inhibition, cancer cells are unable to continue their replication as they simply do not have enough of CMG helicase reserves and therefore go into apoptosis or cell death. And normal cells, however, with sufficient amounts of resting CMG helicases are able to use the reserves and resume DNA replication. Hence, my hypothesis is that RB peptide treatment will only have cytotoxic effect towards the cancerous cell line and leave normal cell lines unaffected. The procedure in this research project involved basic cell culturing techniques to maintain cells viable. When cells reached a confluency of above 90%, these cells were split using trypsin, which helps detach the cells from the bottom of the dish. And a confluency can be described as basically the surface area covered by cells. So if 90% of the dish is covered with cells, then there's a 90% confluency. So then the cells were counted using an automated cell counter after the trypsin treatment, and then plated in a 96 well plate and incubated for a duration of 24 hours before the treatment with different concentrations of gemcitabine and RB peptides started. And these different concentrations were created by diluting from a higher concentration. The duration of treatment was dependent on the time it takes for cells to replicate. It's called doubling time. So for the normal cell line, this was 1 day. So these cells got treated for 2 days in total to ensure a complete replication of all cells in the plate, or 2 doubling times. While for the cancerous cell line, this was 2.5 days. So these cells get treated for the double, so 5 days. And after this, viabilities of treated cells were assessed using a CCK8 assay, and it was put into a plate reader, the 96 well plate, that measures the absorbance of the metabolic activity of the cells, which is like an indirect way of assessing cell viability. Results. So first of all, when I put my 96 well plate into the plate reader, I receive a bunch of absorbance values that I have to process in an Excel sheet in order to make a table that we can make use of and understand. So instead of using these absorbance values, I converted those numbers into percentage using the control that doesn't have any gemcitabine or RB peptide treatment as 100% viable. And as we see for the cancerous cell line, treatment with both the gemcitabine and the RB peptide gives a nice, almost exponential decrease in cell viability as the concentration of the chemicals increase. So the results for the normal cell line. For the normal cell lines, however, we see a decrease in cell viabilities of the gemcitabine treatment, but minimal to no decrease for the RB peptide treatment. This correlates to the theory described above and are some really captivating results. Conclusion. Gemcitabine had equal or very similar results on both cells, as I described above, while RB peptide only had cytotoxic effect on the cancerous cell lines, which correlates with the theory and our hypothesis. This is certainly some really intriguing results and accentuates that our novel approach of using RB peptides could be developed into a chemotherapeutic agent that spares normal healthy tissues and cells and thus invokes less harmful side effects, which we want. So yeah, thank you so much for listening. And I hope you found this interesting. Thank you so much.